Immunopathology. This slide is a medical image of the three main cells of the immune system. The T cell at left, the B cell to the right, and the macrophage center bottom. Notice the multitude of tiny green and red pathogens in the background, how much smaller these cells are compared to the three human cells. During this presentation, we will review the immune response, the cells of the immune system and the antibodies, hypersensitivity reactions, transplantation and blood transfusion, autoimmune and immunodeficiency diseases, and amyloidosis. The immune response includes two complex interacting systems. The first is natural innate immunity, which is relatively primitive and non-specific. It involves mechanical and chemical barriers, along with the various cells of inflammation that were covered in the previous chapter, which recognize and react to any antigen. It also includes the acquired or adaptive immunity. This is found only in vertebrates and is activated when innate immunity fails. This system is acquired through previous exposure to specific antigens. The first exposure activates the body's lymphocytes, T and B cells. Upon subsequent exposures, the immune system is able to recognize the specific antigen and mount stronger and stronger attacks. To function properly, acquired immunity requires the ability to distinguish self from non-self. The body's innate protective mechanisms include the mechanical barriers of the skin and mucous membranes, and chemical barriers, such as lysozymes found in tears and intestinal secretions, along with additional protective proteins, such as complement, found in tissues and blood plasma. It also includes the phagocytic cells, the macrophages and neutrophils, and cytotoxic cells, such as natural killer cells, of the inflammatory response. Taking a closer look at the skin, we can imagine three lines of defense against injury and infection. The first line involves these mechanical and chemical barriers. The second involves inflammation. And the third involves the specific immune responses of acquired immunity. All the cells of the immune system originate from stem cells in the bone marrow. They are all white blood cells or leukocytes. Leuko just means white in Greek. But they develop along two main lineages, as we can see here. One includes the cells primarily involved in innate defense, or the inflammatory response. That is, the neutrophils, the monocytes that become macrophages, and the eosinophils and basophils. The other lineage includes the lymphocytes, those primarily involved in adaptive or acquired defense. These are the B cells that become plasma cells and the T cells. Pluripotential stem cells in the bone marrow develop into all of the various cells found in the blood. Premyeloid cells give rise to the blood cells of the myeloid lineage, which includes those leukocytes primarily involved in innate defense, but also includes the red blood cells, or erythrocytes, and the platelets. Lymphocyte-committed stem cells give rise to three lymphoid cell lineages: T cells, which mature in the thymus, and B cells, which mature in the bone marrow. And finally, the NK cells, natural killer cells. T and B lymphocytes are the primary cells involved in acquired immunity. Natural killer cells mediate innate immune reactions. They do not interact with T and B cells. Some of the leukocytes of the myeloid lineage, however, such as the macrophages and dendritic cells, eosinophils and basophils, however, they might react, and they do react with T and B cells, and they can be considered helper cells in acquired immunity. As reviewed in the previous slide, all lymphocytes originate in the bone marrow. One lineage migrates to the thymus to mature into T cells. Another remains in the bone marrow and develops into B cells. The bone marrow and the thymus are therefore referred to as primary lymphoid organs. From these primary organs, T and B lymphocytes enter the bloodstream and colonize other secondary lymphoid organs, such as the tonsils, hundreds of lymph nodes throughout the body, with prominent clusters in the neck, underarms, groin, and abdomen, and the spleen. Lymphocytes are also found in other organs, particularly the gastrointestinal and respiratory systems, where they are integrated into the mucosal linings. T and B lymphocytes can be distinguished by the type of CD, or cluster differentiation antigens, that are expressed on their surface. The most important T lymphocytes are T helper cells and T suppressor cytotoxic cells. T helper cells, which express CD4, help the B cells produce antibodies. T suppressor or regulator cells suppress unwanted or no longer needed antibody production, while T cytotoxic cells kill infected cells and cancer cells. Both T suppressor and T cytotoxic cells express CD8 on their surface. Along with macrophages and natural killer cells, cytotoxic T cells are involved in what is sometimes called the cellular response. Because the B lymphocytes become plasma cells that produce antibodies, they are involved in what is sometimes called the humoral response. In slide A, you can see the B lymphocyte is dominated by its nucleus and has little cytoplasm. In slide B, the cytoplasm of the plasma cell is well developed, containing numerous stacks of rough endoplasmic reticulum, which are the site of antibody or immunoglobulin synthesis. T cells can only react to an antigen that has been processed, bound, and presented by APCs, antigen-presenting cells, via a particular MHC molecule. These MHC molecules are part of a large family of proteins which are found on the surface of APCs and most other cells as well. There are two groups of MHCs. Type 1, which reacts with CD8 on T suppressor cytotoxic cells. This is found on most cells of the body. Type 2, which reacts with CD4 on T helper cells, which then helps the B cells to produce antibodies that match a particular antigen. Type 2 is found only on the macrophages and lymphocytes that function as professional APCs. This diagram illustrates how antigens are processed and presented by professional APCs using type 2 MHC molecules. First, various antigen proteins are internalized into endosomes via endocytosis. Second, lysosomes fuse with these endosomes to form a vesicle. Then, third, antigen proteins are digested, broken down into smaller peptide fragments. Fourth, a specialized organelle that contains the type 2 MHC molecules fuses with this vesicle, and the antigen fragments are rearranged and bound to MHC. Fifth, the type 2 MHC antigen complexes are exported to the cell surface for presentation to CD4 T helper cells. This figure summarizes the process whereby a B cell is activated to develop into plasma cells. B cell receptors recognize and bind to an antigen. The B cell functions as an APC in this case. It engulfs, processes, and presents this antigen, cradled within the arms of its own MHC molecule, to a helper T cell. The T cell activates the B cell, causing it to differentiate into plasma cells. Every plasma cell is essentially a factory for producing an antibody tuned to a particular antigen. Each of the plasma cells, descended from a given B cell, manufactures millions of identical antibody molecules and pours them into the bloodstream. Antibodies are proteins of the immunoglobulin class that react with antigens. All are composed of light and heavy amino acid chains. The light chains are the same for all the IGs. The heavy chains, designated gamma, mu, alpha, epsilon, and delta, are specific to each of the 5 IGs. IgG is the main defense against infection. It's the most abundant Ig and the only one that can cross the placenta. IgM fights blood infections and helps trigger production of additional IgG. It reacts to blood group antigens and leads to the agglutination of red blood cells. Immunoglobulin A is the major antibody found in the membranes of the respiratory and gastrointestinal tracts, the second most common Ig found in tears, saliva, mucus, and colostrum. IgA is important in local immunity. IgE is found in the mucous membranes and skin. It is the antibody that triggers allergic type 1 hypersensitivity reactions. IgD is present in the blood serum in tiny amounts. It's found on B cell surfaces where it is used as a receptor for antigens. It is the least understood antibody. Each chain has a constant Fc portion and a variable Fab portion. The variable part binds to different antigens. This is a diagram of a generic immunoglobulin molecule. If we outline the heavy chains in dark blue and the light chains in light blue, we can see the familiar Y-shaped symbol that is used to represent an antibody. Antibodies that bind to soluble antigens dissolved in the blood plasma form antigen-antibody complexes that remain in circulation. Like snowballs, these complexes tend to enlarge until they become filtered out by the spleen or liver and phagocytized by macrophages. Smaller complexes may remain in circulation for a long time. Antibodies that bind to insoluble antigens can also coat bacterial cell surfaces in a process called opsonization. This binding may neutralize antigens. Antigen-antibody complexes may also fix and activate complement, promote agglutination, or tag pathogens and recruit phagocytic or cytotoxic cells. In this figure, we see antibodies binding to antigens on the surface of red blood cells, which causes the red blood cells to stick together and form clumps, separating them from the blood serum in the process termed agglutination, which is basically Latin for glued together. On the right, we see an antibody activating the complement cascade, which leads to MAC-induced perforation of the cell membrane and terminates in cell lysis. This animation shows opsonization, how antibodies can stop harmful pathogens from attaching themselves to healthy cells in the bloodstream. It begins with normal red and white blood cells flowing happily through the bloodstream. Next, a single pathogen appears on screen, slowly moving toward its destination on the surface of a cell. The tubular extensions on the pathogen are surface antigens. These attach to corresponding surface proteins on a white blood cell or leukocyte. As the animation continues, more pathogens continue to attach to the white blood cell, rendering it ineffective. During the immune system response, Y-shaped antibodies begin to attack the pathogen, binding to its surface proteins as the pathogen attempts to anchor to the blood cell. Opsonization thus completely blocks the pathogen from attaching to the white blood cell. It also tags the pathogen so that a macrophage appears on screen, recognizes, engulfs, and digests the pathogen. Given all this complexity, it is not surprising that sometimes the normal immune system will overreact, become hypersensitive. Immune system overactivity can lead to undesirable reactions that range from the uncomfortable to the fatal. Injury caused by hypersensitivity results from interaction between antigens and antibodies, or between antigens and sensitized T lymphocytes. When antibodies are involved, the reactions can be considered immediate hypersensitivities, and further classified as type 1, type 2, or type 3. When T lymphocytes are involved, the reactions are characterized as delayed or cell-mediated hypersensitivity. This is type 4 hypersensitivity. All immediate hypersensitivity reactions begin with first exposure, which sensitizes the individual to a particular antigen, such as pollen. The antigen is taken up by macrophages, presented to B lymphocytes, which are stimulated to differentiate into IgE-producing plasma cells. These IgE antibodies fix themselves to the surface of mast cells, where they await second exposure. Upon subsequent exposure, the offending antigen reacts with these surface antibodies, triggering the release of nasty mediators, like histamine, from the mast cell. The end result? Vessels dilate, smooth muscle contracts, and inflammation sets in. The most important clinical manifestations of type 1 hypersensitivity include local atopic reactions characteristic of hay fever. They also include the more systemic reactions characteristic of asthma, atopic dermatitis, and anaphylactic shock. In type 2 hypersensitivity, antibodies, IgG or IgM, bind to antigens on a cell's surface, that being any target cell. Along pathway A, complement is activated, which leads to cell lysis. Pathway A is characteristic of an acute hemolytic reaction caused by a blood transfusion or of hemolytic anemia, both of which result in the lysis or destruction of red blood cells. Along pathway B, target cells are opsonized. Macrophages, natural killer cells, or other effector cytotoxic cells attach to the protruding end of the Ig molecules, the Fc portion, and destroy the target cells, coated with antibodies. This pathway is characteristic of Goodpasture syndrome, which damages tissues in the kidneys and lungs. Finally, not shown in the diagram, the antibodies may react with receptors on specialized cells and stimulate or inhibit their function. This is what happens in Graves' disease, where antibodies bind to receptors for thyroid-stimulating hormone and cause thyroid cells to overproduce hormones. And in myasthenia gravis, where antibodies bind to receptors for acetylcholine in muscle cells and inhibit their function. Type 3 hypersensitivity is also known as immune complex hypersensitivity. In general, antigen-antibody complexes form and circulate, get stuck in vessel walls and the semi-permeable membranes of various tissues, and stimulate inflammation, the end result being inflammation-mediated tissue damage and necrosis. The reaction can take hours, days, or even weeks to develop, depending on whether or not there is immunologic memory from previous exposure. The reaction may be general, as in serum sickness, which occurs when the ratio of antigen to antibody allows soluble immune complexes to form that are too small to get phagocytized, yet big enough to get trapped where plasma is filtered through tissue basement membranes. Or the reaction may be restricted to individual organs such as kidneys, as in post-streptococcal glomerulonephritis. Or it may affect blood vessels, as in polyarteritis nodosa, which can occur as a result of vaccination, when it is referred to more specifically as the arthros phenomenon. In this type of reaction, immune complex deposition and associated acute inflammation in vessel walls leads to the accumulation of fibrin clots in a process called fibrinoid necrosis. A type 3 reaction may also proceed to affect multiple organs throughout the body, as in systemic lupus erythematosus. Type 4 hypersensitivity is the only type not mediated by antibodies. Instead, it is often referred to as cell-mediated because it involves T cells and macrophages. It also referred to as delayed because the cellular response takes from 48 to 72 hours, as opposed to the more immediate hypersensitivity reactions, which generally appear within 12 minutes of exposure to an antigen. In this type of reaction, upon subsequent exposure to a particular antigen, helper T cells secrete cytokines that recruit and activate macrophages and cytotoxic T cells and induce inflammation, which damages tissue. Type 4 hypersensitivity is involved in the pathogenesis of many infectious diseases, such as tuberculosis and leprosy, and can lead to the development of granulomas, chronic inflammatory lesions, that consist of accumulated T cells and macrophages, which transform into epithelioid cells and giant cells. Granulomas can also form in response to tumors and are characteristic of the systemic disease sarcoidosis. Another form of type 4 hypersensitivity is contact dermatitis caused by a variety of allergens, including poison ivy or the chemicals in latex gloves, ointments, antiperspirants. Here we see a clinical presentation of a severe skin reaction to the chemicals in laundry detergent. Granulomas do not form, but note the presence of a large vesicle filled with infiltrates of T cells and macrophages. Transplantation can induce hypersensitivity reactions. Solid tissues can be transplanted successfully only if the donor and recipient are immunologically compatible. Another way to look at this is the graft will take only if the recipient is unable to mount an effective immune response against the introduced foreign antigens. There are several forms of transplantation, including autografts, where the patient is both donor and recipient. Isografts, which is transplantation between identical twins. Homografts or allografts, where the donor and recipient have to be as immunologically compatible as possible. And xenografts, where the donor is not a human, as in pig heart valves. I had a friend actually who had a horse jawbone transplanted. Even so, all homografts elicit some type of transplant rejection through both antibody types 2 and 3 and cell-mediated type 4 hypersensitivity reactions. Transplant rejection is often classified as hyperacute, acute, or chronic. A serious complication called graft versus host reaction is the result of a type 4 immune reaction mediated by transplanted T cells. In this figure, we can see how the reaction to a transplanted kidney involves a combination of hypersensitivity reactions. Antibody-mediated type 2, cytotoxic T cells involved in the development of granulomas through a type 4 reaction, causing tubulointerstitial injury, and fibrinoid necrosis, a type 3 reaction. Blood transfusion is transplantation that involves liquid tissues. The major blood group antigens of types A, B, and O have natural antibodies against them that trigger a type 2 hemolytic reaction upon transfusion between groups. Group A contains anti-B antibodies, B contains anti-A antibodies, O contains both, and AB contains neither. As a result, persons with O blood are called universal donors, and those with AB are called universal recipients. To avoid a hemolytic reaction, cross-matching is important before transfusion. Such cross-matching can also identify potentially problematic minor blood group antigens. The compatibility between major blood groups determines the outcome of transfusion, as indicated in the following figure. Another group of blood antigens called Rh factors can cause incompatibility that results in a hemolytic type 2 hypersensitivity reaction. But because Rh antigens, of which the D antigen is the most significant, have no natural antibodies, previous exposure is required to develop immune sensitivity. If your blood contains these D antigens, you are called Rh positive. If a and the father Rh positive, the fetus may be Rh positive. During delivery, the fetus's red blood cells can mix with the mother's blood, cause her to produce anti-Rh, specifically anti-D, IgG antibodies. Upon subsequent pregnancy with an Rh positive fetus, the Rh negative mother is now sensitized, and her preformed IgG antibodies will attack fetal red blood cells. Hemolysis may result in erythroblastosis fetalis. Such an outcome can now be avoided if Rh sensitization of the mother is prevented through timely injection of anti-D immunoglobulin using a medication called RhoGAM. Rh incompatibility is probably the most important maternal-fetal blood type incompatibility. Clinical features of Rh factor incompatibility range from the relatively mild to the fatal. For example, with icterus gravis, the only manifestation of hemolysis is jaundice. Occasionally, however, a complication may develop termed kernicterus, which can cause permanent brain damage. With erythroblastosis fetalis, the fetus becomes anemic and develops severe hypoxia and congestive heart failure. Fetal bone marrow works overtime, trying to compensate for the loss of red blood cells. Other organs are recruited for the production of red blood cells, such as the liver and the spleen. This may progress to hydrops fetalis, in which the fetus dies in utero. Autoimmune disease is the result of an abnormal response to self-antigens. A healthy immune system is able to distinguish between self and non-self. Our body's cells all express antigens, yet our immune cells normally remain tolerant of these self-antigens and react only to foreign antigens. Over millions of years, numerous control mechanisms have evolved to keep our immune cells from responding against self-antigens. But sometimes these mechanisms fail, and an autoimmune disease develops. To diagnose a disorder as autoimmune, look for the presence of autoantibodies in the blood. Evidence that immune mechanisms, hypersensitivity reactions, have played an important role in its pathogenesis and may be the cause of the distinctive pathologic lesions. Also, indirect evidence of an immune nature. For example, immunosuppressive drugs such as steroids help. It is unknown what causes the immune system to attack itself. Autoimmune diseases occur with more frequency in some families, which suggests there may be a genetic predisposition. Some autoimmune disorders are linked to certain human leukocyte antigen haplotypes, HLA. A haplotype is simply a group of genes inherited together from a single parent. These HLA genes, then, are simply the human version of the MHC genes found in most vertebrates, which code for particular MHC molecules. Because many autoimmune diseases are more common in women than men, non-genetic factors may be important as well. Autoimmune diseases can be systemic or organ-specific. If systemic, they involve multiple organs. Common systemic autoimmune diseases include systemic lupus erythematosus, SLE, which can be identified by its distinctive butterfly rash, rheumatoid arthritis, and polyarteritis nodosa. Those limited to single organs include multiple sclerosis, which attacks the central nervous system, Hashimoto's thyroiditis, and Graves' disease, which attack the thyroid, and autoimmune hemolytic anemia, which attacks the blood, which is not a tissue. Which is not an organ, it's a tissue, but also myasthenia gravis, which attacks the muscle. While autoimmune diseases are caused by an overactive immune system, immunodeficiency diseases are the result of an underactive immune system. They can be primary or congenital or secondary, which means they are acquired. There are many primary immunodeficiency diseases, but among the most important are severe combined immunodeficiency, which results from a defect in the lymphoid stem cells, such that T and B cells may never develop. There's also an isolated deficiency of IgA, which is the most common, yet it's often asymptomatic. Finally, there's DiGeorge's syndrome, which is caused by a chromosomal defect which affects the thymus and leads to T cell deficiency. Acquired immunodeficiencies can result from malnutrition, aging, and the use of particular medications, such as chemotherapy, immunosuppressive drugs, steroids. Many diseases can also directly or indirectly cause immunosuppression. But acquired immunodeficiency is, of course, characteristic of acquired immunodeficiency syndrome, or AIDS, which is caused by the human immunodeficiency virus, HIV. HIV directly infects T helper cells. This retrovirus also infects and is stored in macrophages and related phagocytic cells, such as those found in the lymph nodes and nervous system. This 3D medical animation shows an HIV particle releasing its viral core or capsid into the cytoplasm of a T helper cell during viral replication. Once inside the T cell, the capsid disintegrates, releasing viral RNA. The viral RNA and reverse transcriptase enzyme begin creating a new strand of viral DNA using the viral RNA strand as a template. This is why it's called a retrovirus, by the way. Ultimately, the T cell dies and falls apart through apoptosis. Watch it explode there, which sends more copies of the virus into the bloodstream. During the course of continued viral replication, more and more T cells are thus destroyed. Once infected with HIV, there are 4 recognized phases of the disease process. There's the early phase of acute illness, which only occurs in about 50% of cases, and chronic phases of asymptomatic infection, which may or may not be accompanied by a phase of persistent generalized lymphadenopathy. The crisis or terminal phase of AIDS is characterized by superimposed infections and complications. AIDS is a systemic disease that can lead to pathologic changes throughout the body. Immunodeficiency leaves the patient vulnerable to various opportunistic infections of the lung, the gastrointestinal tract, and the central nervous system. The malignant tumors that can develop in AIDS are important causes of death. AIDS patients show an increased incidence of all tumors, but especially lymphomas of the lymph nodes or GI tract, and lymphomas of the central nervous system. Also a distinctive skin tumor called Kaposi's sarcoma. Finally, amyloidosis is a group of diseases in which one or more organ or organ system accumulates deposits of amyloid, an abnormal protein defined by its physical rather than its biochemical properties. These amyloid deposits often develop as an abnormal immune or inflammatory system response. Amyloid forms distinctive, thin, nanoscopic fibers which arrange themselves into sheets. It can be detected because it stains a certain way with Congo red. Biochemically, there are many forms of amyloid. The most important 2 groups are AL amyloid and AA amyloid. This figure summarizes the pathogenesis of amyloidosis secondary to inflammation. Cytokines released from inflammatory cells stimulate the liver to produce serum amyloid precursor, or SAA. This soluble precursor to amyloid enters the bloodstream, is processed by macrophages, and deposited along the tissues in those distinctive fibrous sheets. The clinical presentation of amyloid deposition depends upon the organs or organ systems affected. For example, deposits of a particular form of amyloid in the brain are associated with Alzheimer's disease. Systemic amyloidosis is usually caused by deposition of AA or AL amyloid throughout the body in various organs such as the liver, kidneys, adrenals, spleen, and the heart. Wow, well, that's about it for everything we need to know about immunopathology. Thank you for watching. It's been a pleasure. Have a lovely rest of your day. Thank you. Bye.